Right, so we are now being recorded. So welcome to this 8th video teleconference for BIOL 105 YC. Um, there is a pending quiz that's open for you right now. If you haven't taken the quiz, the content of the quiz will be what we're going to review this morning. So I suggest that you really interact no, with this particular video teleconference. In terms of schedule, let's look at the schedule. Um, we have another lesson supposedly before the semifinals, but I'll tell you ahead now, the coverage of the semifinals now will be from chapter 7, and then we have chapter 8, and then we have chapter 9. Teka muna na, wala yung presentation. Okay, so the coverage for the semifinals will be chapters 7, 8, and 9. I will not include this genetic information part. In fact, I'm planning right now, and I haven't told the other sections yet. Now, for the genetic information portion, I will not be posting lessons next week siguro. Maybe we can still meet next week for a video teleconference, and then I will be giving your exam on the last week of March as seen here. So maybe not on the 25th to 27th, so that we can still meet and review for chapter 9 on that those days. Right. So with that said, let us review this morning chapter 8, okay, cytoplasmic membrane systems or the endomembrane systems of the cell. Okay, so this is quite a long chapter, and I've asked the other groups as well on how they are doing with the reading, no? Not just for this chapter, but for the other chapters, but most particularly now up to now. How are you with your reading? Are you understanding the contents? Do you have difficulty uh, in your reading schedule, etc.? May I hear from you?
Yes, sir. So far, okay ra sa ako, sir. And then, kaning sa chapter 9, kay nanako dapit sa mga last parts about sa katong actin filament, sir.
All right. So no, so chapter 9 naman ta karon, but this is a review of chapter 8. So that's good to know for you. What about the others? Any challenges or difficulties that you met no along the way while studying? What's the most challenging part? Silence means maybe you are doing fine. You are agreeing with the first person who shared na you're able to keep up with the readings. Okay, so just message me if ever there is a problem. But of course, the purpose of this video teleconference is to help you review, or help you realize, oh, I don't know about that portion pala of the lesson. So let's start. Okay, so I have here an illustration, a general view of the endomembrane system of the cell. Can anyone from the group tell us something about this illustration?
Hello, sir.
Yes.
For this illustration, in general, there are two types of movement in the endomembrane system, particularly anterograde direction, which is towards the plasma membrane, and retrograde direction, um, which is towards the rough ER, sir. And for anterograde direction, there are certain protein coatings, particularly COP2 proteins, which handle the movement. And for retrograde direction, the protein coatings responsible for it is the COP1 protein coating, sir.
All right, so everything that you've said is correct. Now, so let me just admit someone in. Okay. So everything you said was correct about this illustration. Okay, so there are two movements as you mentioned already, and they are both equally important. No, so the vesicles need to move. Why? Because our endomembrane system of the cell is not just the factory. No, the ER is the factory, but it's also a delivery packaging system. No, like LBC, and there needs to be delivery of materials made in the ER. So the delivery can either go both ways in direction, anterograde or retrograde. And you mentioned coating proteins, no? That's actually the meaning of COP. COP. COP means coating protein. And then there are two of these COPs. We have COP2, which coat the vesicles that move from the ER to the cis Golgi network, no? In that direction alone. And then we have COP1, coating protein 1, which coats vesicles moving from the trans Golgi network in a retrograde direction back to the medial or the cis Golgi network, or even back to the rough ER. And then we have clathrin, no, which has a different naming scheme. Clathrin coats vesicles that move from the TGN, as we see here, towards the cell membrane. So I have a question: true or false? All vesicles that move in a in an anterograde direction are coated by COP2. All vesicles that move in this direction are coated by COP2. Is that true or false?
Somewhat false, sir,
Why?
because there's also the clathrin coating proteins that deliver from the TGN to lysosomes, sir.
All right. So that is correct. So correct reasoning. No? So we also have these secretory granules which don't even have coating proteins at all. And these clathrin, like COP2, can coat vesicles that move in an anterograde direction. So you are correct. Another one, true or false? All vesicles that move in a retrograde direction are coated by COP1. True or false? Anyone?
I think true, sir,
Based in this illustration,
based on
true.
this illustration.
But
Para sa kuha, sir, kay kuan not necessarily true gyud kay
okay,
naapoy ano ito, clathrin proteins that are involved in the ano ito gani housekeeping receptors pod for ligands, sir, and they sort of coat those vesicles with the housekeeping receptors, sir, towards endocytosis pod, sir.
okay. So based on this illustration, you might say, oh, only COP1 coats retrograde moving vesicles. But the answer actually is false, no? So as mentioned by Mr. Coronia, um, the clathrin, not shown here, can also coat vesicles that move from the surface of the cell to the inside. No, clathrin also does that. So the same as COP1, clathrin can coat vesicles that move in a retrograde direction. And the same as COP2, clathrin can also coat vesicles that move in an anterograde direction. So cyclothrin in both directions. The only difference is clathrin may only be found somewhere here between the TGN and the cell membrane. No, there are no clathrins here in the ergic zone. No, the ER, Golgi, what's the I? Intermediate compartment. So there are no clathrins here, only COP2 or COP1. No, and COP1 can also be seen here. So please take note of that. That might come out of the quiz. So here in this illustration, I want us all to focus on this receptor, no, that is pointed here. Anyone tell us what is the function of this receptor as depicted by this figure? So you look at the sequence here. Sorry. Look at the receptor. How is it depicted? What is it doing?
I think here, sir, only the purple proteins are being selected by the receptor, which is this one you can see. So on the second segment, there's a vesicle that is being pinched off, and this receptor somehow guides it to dock into another endomembrane compartment or organelle. And then so I know, sir, this vesicle on the end part then fuses with that organelle, and so the purple proteins are then transported in that lumen or inside of the endomembrane organelle. So by that we accomplish na tanan diri sa taas are all purple proteins lang, and then we left behind the white proteins on the original organelle, sir.
Correct. Okay, so everything about that is correct, no? So it's just very visual, no, that the book actually uses different colors for the different proteins. So katong purple proteins that you mentioned are the soluble proteins or the secretory proteins, no, meaning to say the cell is going to be secreting them later on. So they are the only ones selected by this special receptor, so that they are the only ones that are carried through inside this vesicle to be moved into another compartment, as you mentioned. So these white colored proteins or light colored proteins here are known as the resident proteins, and they are not included inside the vesicle because the receptor doesn't recognize them. So can you give an example of a protein that needs to stay where it's supposed to stay, like this resident protein?
ER resident protein, sir.
Okay. So particularly enzymes, di ba? So enzymes. Enzymes, the function is to catalyze reactions, and they need to be in a particular place. They don't need to be moving all around all the time. No, so they stay where they are supposed to stay. So this is their office. No, ito the one at the bottom. So they are resident proteins on this area. So if an enzyme happens to be wrongly moved into this another compartment, there has to be a way to return it back to its original location where it's supposed to be. So enzymes are examples of that. And your ER resident protein, those are most likely enzymes as well. Okay, so it's very important to see this one. The coating proteins are not shown here. Okay, so take note, it's the receptor proteins that bind to the selected soluble proteins, which are the secretory proteins. So wala gipakita diri ang coating proteins. No, not yet. So later we'll see an illustration with the coating proteins. So let's talk about the first organelle that is included in the endomembrane system, and that's the ER. There are two types. So anyone from the group want to generalize on the structure, the function, the types of the ER?
Sir, for the endoplastic reticulum, there are two types. First is the rough ER and then the smooth ER. The rough ER is named rough because it is covered with ribosomes, whereas the smooth ER is not. And in general, the rough ER has like this um, stacking pattern of cisternae structure, and for the smooth ER, it's more like it looks like tubular structures, sir. And then the function of the ER is to um, it's like a factory for most of the protein synthesis and lipid metabolism, sir.
All right, so you are correct. No, again, let me just add to the function. The rough ER is for protein synthesis, no? So it's a factory talaga. The smooth ER is also a factory, but because it lacks ribosomes, it's not really for protein synthesis, but instead it specializes in lipid synthesis, no? So lipid metabolism, as you mentioned, particularly lipid anabolism, so the formation of new lipids for the cell. All right, so this is the factory, no? So just backtrack a bit, no? Everything that we're talking about here, the endomembrane system, is only true for eukaryotic cells, no? Prokaryotic cells do not have the endomembrane systems. So this is the factory, and this is the part of the cell that a virus would hijack, no? So the virus will take control of the factory of the cell, which originally produced lipids and proteins for the cell. But when the virus invades this, the viral DNA will now dictate for the production of viral proteins instead of the cell's proteins. Right. So this is a detailed view on how the protein, particularly secretory protein, is synthesized in the rough ER. Okay, so we have a sequence here, but we don't want to talk about each one in very detailed manner. So can anyone from the group summarize to us what's happening in the stages here?
Go on, sir. First, the ribosome carrying the mRNA and the nascent polypeptide sort of approaches this the ER membrane, and this signal recognition particle, or the SRP, this brown color thing, attaches to the nascent polypeptide of the ribosome. And with the help of the SRP receptor, the ribosome is able to sort of dock into the membrane, and then it attaches itself to the translocon, and then the SRP detaches itself. And then for the last part, the nascent polypeptide is now being chaperoned by this chaperone in the lower right corner here.
All right, so those were correct. No? So the role of the chaperone protein here is to help this nascent polypeptide to fold properly. No? So the polypeptide really enters the ER lumen, no, because it's destined to be secreted. No, the purple proteins kanina na sinabi ni Miss Adaptar, no, the ones that are held by the receptor proteins, this is an example of that, no, a secretory protein that really enters the ER lumen. Okay, but then there is another type of protein, no? These are general types of proteins, not specific types. the integral membrane protein. So this protein is destined to be part of the cell membrane. So look at the process on which it is incorporated in the ER membrane. It's a different process from the secretory protein. So can anyone from the group summarize to us what's happening in the steps here?
In here, sir, the difference mostly lies on the nascent polypeptide not going all the way into the lumen. Uh, since previous is dito sa synthesis is naano jud siya diba sir ni go siya all the way. So in here, certain charges affects its composition or the amino acid composition, and it causes it to orient at a particular segment lang to align sa membrane's thickness or orientation. So here, the translocon is capable of, as you can see, is opening sideways. So the nascent polypeptide then floats laterally to it. So that's why na-integrate gid siya dito ang membrane ang protein sa membrane. So also in here at the right side, farthest right side, we can see that the orientation is also somehow reversed. Nadayon siya, sir.
Okay, everything about that is correct. So the orientation, as you mentioned, no, is really based on the sequence of amino acids and which portions have the positive or more negative charge in the nascent polypeptide. Because as you know, in the previous lesson, diba, the cell membrane has a membrane potential. Usually, it's more negative on the outside than the inside. So ang nascent polypeptide pod might have its own charges, and so it orients itself based on the attraction of positive to a negative charge. Okay, and then yes, the translocon is actually a complicated machinery because it's not just a channel; it can also open a hinge sideways, so that as you mentioned, the integral membrane protein will just diffuse towards the membrane here of the ER. Okay, so this is synthesis of integral membrane protein versus synthesis of a secretory protein. Okay, so now that we sort of know the steps no on the synthesis of these proteins, okay, before that, let's look at here what really happens next to our integral membrane protein. So this is integral protein, like this one. Okay, look what happens now in the rough PR. You can see that the protein has two ends. No, we have an end here that's found in the lumen. It contains these Y-shaped orange things. What are these?
Oligosaccharides, sir.
All right, these are oligosaccharides. And notice when it's carried in the vesicle, notice that the oligosaccharides stay with inside the vesicle. And when it fuses into the Golgi complex to be packaged, the oligosaccharides remain in the lumen. And then it's carried by another vesicle again. No, this one could be COP2 coated, no? And then this one here, the vesicle here on top could be clathrin coated. So notice the oligosaccharides are still inside, but once the vesicle fuses with the cell membrane, the oligosaccharides end up to be in the exterior part of the cell, no? So motong glycocalyx dayon in our previous lesson. So this one is the pattern for the movement of integral membrane proteins, while the illustration a while ago, this one is for secretory proteins, no, soluble proteins. So you need to be able to distinguish and describe the differences of these two. Okay, so now we go to the process of glycosylation. How is that oligosaccharide added to your protein. Anyone wants to summarize all of these steps? So this is in the ER still, the glycosylation or adding of oligosaccharides. Which molecule carries the oligosaccharide chain first before transferring it to the nascent polypeptide? Anyone? Kinsa ang una magdala sa oligosaccharide chain?
The dolicol phosphate, sir.
Okay, so that dolicol phosphate, no, we won't discuss the detail anymore here, no? Pero we notice there is this uridine diphosphate, you have monophosphate, you have here GDP, murag ADP, no? This is the guanosine diphosphate, etc. We will not focus on that now. Instead, let's just see how the dolicol phosphate actually carries first the growing oligosaccharide chain. So first, i-add sa iya will be the NAGs, no? And then later on top of the NAGs will be the mannoses, no? And then here's a weird, not weird, here's a thing to note. This dolicol phosphate is capable of flip-flopping, no, magbali-bali. So it will reverse its orientation so that your oligosaccharide is now on the inside of the rough ER, no? And then the oligosaccharide chain continues to build up. So nara imong mannose. And here's very important part, ang pinakatumoy will be 3 glucose molecules. And once this is completed, it is then transferred to your nascent polypeptide here, no, from your ribosome. And this is now the glycosylation of your polypeptide or protein. Okay, so that's the process basically. So question, what is the function of these last 3 glucoses? If you can remember.
It's for quality control of misfolded protein, sir. If I remember, it is being used as sort of like a ticket for Calnexin, sir.
Okay. So this morning we will have another sort of analogy. So kaning nga process, glycosylation is like if you still remember the days when there was no lockdown and it was almost exam week, no? Maglinya mo sa finance office. Okay. So this is like that process where you fall in line in the finance office so that you will pay and then eventually you will get your admit to exam permit. So this glucoses that we see here will be like the admit to exam permit of your nascent polypeptide. So now that the nascent polypeptide got the admit to exam permit, ready na siya for quality control for the actual exam, no? So the polypeptide will now go to the room where the exam is going to be made, no? And along the way, the exam permit is gonna be signed by the proctor. So signan. Okay. Tapos once not just sign pero murag gisian pag yun gamay, meaning to say there's a proof na nagamit na siya. Okay? So after that's done by Calnexin. And then it doesn't mean that you have the permit that you will pass the exam. It doesn't mean that you have the glucoses here that this protein will pass the test. So if ever the protein was found out to be misfolded, then Calnexin will tell it to go back, get another permit in the form of glucose, and then get folded again, and then come back to take another test. And so this will go again and again until such time that the protein is able to fold properly, but there is a limit. No? If the protein is really not able to fold properly, maybe because the primary sequence of the amino acid is mistaken in the first place, then the protein will be sent to its doom. Where is that? Where will the misfolded proteins be sent in this illustration?
To the proteasome, sir, where it will be destroyed, sir.
All right, so the protein will be sent to this proteasome. The proteasome will dismantle the proteins, no? So ang mga amino acids will be removed from each other again, and the protein fragments will be used again by the cell to make new proteins, di ba? And hopefully be able to fold that new protein properly. But good news, if the protein is able to fold properly, it will be sent here in this station where it waits for receptor proteins to recognize it so that it may be sent into a vesicle to be carried to where it's supposed to go. So this is the quality control step now. And the ticket for this would be the 3 glucose molecules added during the glycosylation process in the rough ER. Okay. So, but it doesn't end there. We also have this UPR. This is the unfolded protein response. So, if ever nagpatong-patong ang trabaho, no, na the proteins are not folded properly, we have this for mammals only. So, we have 2 acting molecules here. We have the PERK, and then we have the ATF6. So, can anyone summarize how the PERK helps in the unfolded protein response? Perk first.
For the PERK protein, sir, this BIP chaperone acts last like chaperone, sir, um, for the misfolded proteins, and once it sort of carries away the misfolded proteins, sir, the two PERK proteins become a dimer here in uh, yeah, step 3, and then the translator uh, factor um, sort of becomes phosphorylated by the PERK proteins, and this this translation factor then travels to a ribosome, sir, where it tells it to sort of stop protein synthesis for a while, sir, due to
Okay.
the misfolding.
Yes, so that's correct. Now stop production first because we still have pending work to fold the proteins properly. So what you just described is actually called a pathway. No, it's a pathway. And in cell and molecular biology, there are many pathways, no? Para siyang schematic diagram on how things work inside the cell. No? So, okay, very good. You were able to describe it clearly. I would like to think of these PERK and BIP as they are inactive because they are both nagchismis pa sila. Okay. And then if ever there is an alarm that there are misfolded proteins, si BIP ang tawagon. So tawagon si BIP to help fold the proteins. And so si PERK is now able to focus on its work because wala na to si BIP. And as you described, it will activate a translation factor, but the translation factor actually helps stop additional translation. So this is what we call a negative feedback. No, negative feedback. Kaya nagtapok-tapok ng trabaho, hunong sa. Okay. Next, we have the ATF6 pathway. So again, anyone in the group describe how the ATF6 pathway is like.
For this ATF6 pathway, sir, as you can see here, BIP is also attached to it. So this BIP has the same mechanism as to the PERK. So it helps protein also, protein folding also. And when it's activated na din, sir, the cytoplasmic portion, which is the one that is detached here, um, will go to the nucleus. And this will promote the transcription of mRNA here. So the translated or translation then produce proteins that are capable of alleviating the ER stress, sir, due to the unfolded proteins that are accumulating.
Okay, so correct. No, so this is like your body coping with the stress no of whatever things that are piling up for you to do. So what do you do? So this is one pathway that the cell does it through the ATF6 pathway. And the stressor here is the build up of misfolded proteins. Okay, so very good. Both of you just described two pathways for the UPR, unfolded protein response in mammals. So next organelle, no, mataas yung ER, is the Golgi apparatus. So we notice here the structure. I think you already know. We have several regions of the Golgi complex. We have the cis region, and then we have the trans region. And just like in the ER, glycosylation also happens. So you can see here, no, in number 1, this is exactly where we left, no, in the glycosylation. Tinanggal na yung tatlong glucose kasi ginamit na for the quality control. So the protein here is sort of expected to be properly folded. And the final product of the glycosylation here will add these other carbohydrates, the galactose, the fucose, and the sialic acid. Okay, so let's talk about the mechanism of the movement of materials in the Golgi complex. There are two theories, no, or two models, but only one of them is correct. So here on the left, we have the vesicular transport model, and here on the right, we have the cistern maturation model. So which among these two was proven to be correct based on evidences?
The vesicular transport model, sir, is more correct, sir, because it only uses um, ito gani. Only uses vesicles to transport the cellular materials from the ER to the Golgi and then to the plasma membrane. Whereas this cisternal maturation model on the right, sir, um, it sort of uh, the material sort of kaning passes along this Golgi complex, sir, and kanang as the Golgi complex matures, kay like or siya uh, becomes more absent, sir, in cells, sir.
Right. So I get your point that the vesicular transport model is the one that is proven to be correct. So let me ask you a question. No, Okay, this one shows us, both of them show us the transport of materials from the Golgi to the outside. No, so that's okay. So my question is this: what about certain resident proteins that have mistakenly been brought to a different part of the endomembrane system? How are they returned back to where they're supposed to be? Resident proteins. Can both of these explain how resident proteins are returned? Actually, my question might confuse you because after all, sir, in the previous illustration, you showed us that receptor proteins make sure that resident proteins are not transferred, no, in the first place. But in reality, based on actual studies, no, in the laboratory, they found out that um, resident proteins could actually somehow be transferred into a place where they're not supposed to be. That was found out in the lab. No? So meaning to say the receptor proteins are not 100% perfect in just selecting the secretory proteins. So which among these models show a possibility of returning resident proteins back to where they're supposed to be?
The cisternal maturation model, sir.
Okay, so ito. Okay, so this one can explain going anterograde, and this one can also explain going retrograde. No? So in that case, the cisternal maturation model is actually the one that's favored based on evidence, no? As opposed to the vesicular transport model. So here we can see from in the ergic zone, no, in this direction, we have COP2 coated proteins. Ito pabalik. This is actually the part where certain resident proteins, as the cistern matures, it moves upwards anterogradely, and it carries with it some resident proteins, so they have to be returned back to where they're supposed to be through vesicles coated in COP1. No, and that's based on evidence. And then here we have the vesicles coated with clathrin, or maybe they are um, granule secretory vesicles that do not need to be coated with clathrin, moving from the trans Golgi network to the cell membrane. So again, let me tell you, based on evidence, it's this second model that is proven to be correct. Okay, so are there any questions regarding that or violent reactions? So you read your book, di ba? Meron dong picture talaga na pinakita yung experiment and why that proves that this one is the favored model, or why that shows that this one is the favored model. So it's somewhere there. All right, so let's continue now because we are running out of time. So here, this one is more detailed. No? So you can still see the receptor proteins binding to the soluble proteins, and you have here the resident proteins that are not included. And here we can now see that it's not just the receptor proteins that are involved, but there are also now coating proteins. Now in this case, this is COP2 that covers the vesicle. And this aggregation of proteins, both receptor and coating proteins, causes this portion of the membrane to bulge, now, and eventually pinch off to form this coated vesicle. Okay, so this is now how the vesicle is actually believed to be formed, no? So pag nagtapok-tapok ang mga proteins, mag-bulge din siya, and then it will eventually pinch off the endomembrane. And these are the structures, no, of our coating proteins. Clathrin and COP1 have this triskelion shape, while COP2 is sort of linear. Okay, but notice they have their own subunits. No, there are several subunits for COP2 and COP1 at least. And when they are covering the vesicle is going to be shaped like these. No, although we don't see it in the pictures because they are too small. Right, so now I believe we understand this one more. So let's continue. So the vesicles need to move. And to address the question, how do they move? What's the answer? How do they know in which direction they need to go to and what makes them move?
It is through cancer. Motor proteins that attach to the vesicle, which are also attached to the cytoskeleton microtubules, sir.
All right, so vesicles can move in both directions because if you already read no in the previous in the chapter right now, there are motor proteins for each direction. No, in the case of microtubule, we have the kinesins which can move from the negative pole to the positive pole, and then we have the dyneins which can move from the negative pole to the positive pole. And in case of actin filaments, we have the myosins. No, we have the myosin 5s and myosin 6. Okay, so this is how vesicles are moved. Now, with the help, of course, of ATP. And then when vesicles reach their destination, they are going to be docked. Tethered first. So this is like dropping the anchor of a ship, no, because the ship has already reached the port. So nag-anchor na siya. So you have the tethering proteins here, no? And then once tethered and secured, there will be the docking. So this one, ginaog na din to ang ladder, no, para makanaog na mga pasahero. And the ladder here is in the form of snare protein. So you have the V snare for vesicle snare, and the T snare for target membrane snares, no, to dock the vesicles. And after this, the contents of the vesicles can then move into the new compartment here at the bottom. Right. So here, we have a specific type of protein that is destined to go to the lysosome, no? What is that signal that is found on this specific type of protein na kabalo man ang cell nga dapat sa lysosome siya padulong?
The signal is in the form of mano 6 phosphate, sir,
All
which is
right. So
present in the lysosomal enzyme, sir.
yes, so lysosomal enzymes destined to go to the lysosome, of course. In their oligosaccharide chain, there is a mannose-6 phosphate. And a mannose-6 phosphate receptor recognizes that and will only attach to those types of proteins. And so a vesicle is formed, and this time it's clathrin coated, no, and it will be carried into a direction so that it will become a lysosome. So you see here the clustering proteins disassemble, they go back to where they're supposed to be. The vesicle returns back to where it's supposed to be while the protein reaches its destination. We also see here endocytosis wherein certain foreign bodies might be engulfed by the cell so that it can be digested in the lysosome. And then here we have a process called um, what's the process called here? Autophagy, no, or autophagy, wherein the cell actually needs to dismantle and destroy old organelles, like this mitochondrion surrounded by this phagophore here, which then merges with the lysosome so that the old organelle is destroyed, and in replacement, a new organelle will be made. Now, so this is maintenance of the cell. Old organelles destroyed, new organelles made. Okay, and then here, this one is very important. We have two receptors waiting here outside. Now we have a signaling receptor in green and a housekeeping receptor in red. So notice they carry different ligands going in. Now, an example for the housekeeping receptor ligand here is the LDL, no, the bad cholesterol. It's carried inside, An example of the ligand for your signaling receptor is perhaps a hormone, no, a signaling molecule, a hormone. So notice here that the signaling receptor ends up getting destroyed in the lysosome, while your housekeeping receptor ends up returning back to the surface. So question, why does it have to happen this way? Why should the signaling receptor be destroyed and the housekeeping receptor be sent back?
Um, the signaling receptors are destroyed, sir, in order to avoid the cells being overwhelmed by hormones and other signals, sir. And this process is known as receptor down regulation, sir. And yes, sir.
All right. So I liken this to someone who's always calling you for the nth time. So eventually, now you know that they're calling you, but eventually you are disturbed by that. You want that to stop, so you maybe turn off your phone or put it in silent. So that this is the way of the cell of putting the signaling receptor in silent. It destroys them, okay, because it already received one message. One message is enough for a Y cell. So next, why should the housekeeping receptor be returned back?
They are being returned back or recycled, sir, in order for them to continue on sort of cleaning the outside of the cells from LBL and other bad cell stuff, sir.
Right, that's correct. Now, so the term palang housekeeping, it's really similar to what we how a housekeeper. Now, a housekeeper comes back here and again, now and again, in to maintain the cleanliness of the house. For example, di ba? Pag nag-hire ka ng housekeeper, eto, papupuntahin mo siya sa bahay niyo para maglinis. After he or she is done, okay, uwi, no? Balik, and then you call that housekeeper again to do the job. So you don't destroy the housekeeper. You want them to live, continue, continue existing to be able to continue doing their job, which is housekeeping job. Okay, so very good. So I think we all get that one. So this is just a summary of phagocytosis. No, this is large chunks of materials drawn into the cell. Then we have endocytosis, which is smaller molecules that are drawn into the cell. And then pinocytosis, wherein the cell sort of drinks small amounts of extracellular fluid. And all of them lead to the lysosome where they are digested, and here we even have autophagy, no, the recycling or of old organelles by the cell itself. So lastly, of course, we have the post-translational uptake of proteins. And peroxisomes, the proteins are delivered whole. They don't need to unfold. They just enter the translocon of the peroxisome directly. But in mitochondrion and chloroplast, the proteins have to unfold first before entering these organelles. How many destinations are possible for the mitochondrion?
Four possible
Please
destinations, sir.
enumerate.
For the first one is for the outer membrane, then the intermembrane space, the inner membrane, and of course the matrix, sir.
All right, so those are the possible delivery points of the proteins. In the chloroplast, how many possible locations?
We have six here in the chloroplast, sir.
All
The
right.
outer membrane, intermembrane space, the inner membrane space, and then the stroma and the thylakoid membrane and thylakoid lumen.
Okay, so that's just the inner membrane and not the inner membrane space. Maybe you were talking about the intermembrane space. Okay, so good, no? So please remember those destinations, no, and the mechanism of how the proteins go inside these organelles. Okay, so the thing here is the proteins are not made in these organelles. They're made in the ER. So they have to be delivered in these organelles. And that's why we have the post-translational uptake of proteins. Okay, so reminders, the quiz is now open. It will be closed on March 24, 6 p.m. No, Wednesday next week. So this quiz is about what we talked about this morning. So please continue reviewing if there are still things you need to review. And group discussion has been posted, so I hope you're done posting in your group discussions. So just keep informed, or just check the course page every now and then for the new quiz for chapter 9 and the new group discussion for chapter 9. And again, our exam is going to be moved at the last week of March, no, so that we can still meet next week for another video teleconference. Right, so with that, we end our video teleconference number 8. Are there any questions or concerns? Okay. If you don't have any questions or concerns, or if you have questions later, of course, you can always message me. So I thank you very much, guys, for joining this video teleconference. It's nice listening to you. You're the one discussing, meaning to say you learned, no, from what you've studied. So continue that. Continue being observant with the figures found in the book, because it's very important to understand them. Okay. So thank you very much. Without you, this will be impossible, no? So, dili ta maka-record og video teleconference kung wala mo nag-attend. So have a productive day ahead, and you are now all dismissed.
Thank you, sir.
Salamat, sir.